Hi, people. We're continuing with life science and cell biology in a nutshell. We're continuing reading my notes here. We left off at the organelle called peroxisomes, which contain oxidative enzymes such as catalase and urate oxidase at such high concentrations in cells, in certain cells more than others, that peroxisomes stand out in electron micrographs because of the presence of a crystalloid core. Because of these strong enzymes, which are acidic, like mitochondria, peroxisomes are major sites of oxygen utilization. One hypothesis is that peroxisomes are a vestige of an ancient organelle that performed all the oxygen metabolism in the primitive ancestors of eukaryotic cells. When oxygen produced by photosynthetic bacteria first began to accumulate in the atmosphere, it would have been highly toxic to most cells. Peroxisomes might have served to lower the intracellular concentration of oxygen, while also exploiting its chemical reactivity to perform useful oxidative reactions. According to this view, the later development of mitochondria rendered peroxisomes largely obsolete because many of the same reactions which had formerly been carried out in peroxisomes without producing energy were now coupled to ATP formation by means of oxidative phosphorylation. And the oxidative reactions performed by peroxisomes in present-day cells would therefore be those that have important functions not taken over by mitochondria. Peroxisomes are so named because they usually contain one or more enzymes that use molecular oxygen to remove hydrogen atoms from specific organic substrates, designated here usually as R. The unknown in an oxidative reaction that produces hydrogen peroxide, H2O2. Catalase utilizes the H2O2 generated by other enzymes in the organelle to oxidize a variety of other substrates, including phenols, formic acid, formaldehyde, and alcohol. By the peroxidative reaction, H2O2 plus the R, H2 yields R plus 2 water molecules, H2O. This type of oxidative reaction is particularly important in liver and kidney cells, which are specialized in detoxification. So peroxisomes detoxify various toxic molecules that enter the bloodstream. About 25% of ethanol that we drink is oxidized to acetaldehyde in this way. In addition, when excess H2O2 accumulates in the cell, catalase converts it to H2O through this reaction that we just saw. H2O2 plus a molecule with hydrogens, two H2s, yields just the molecule itself gets dehydrogenated, yielding 2 water molecules. A major function of the oxidative reactions performed in peroxisomes is the breakdown of fatty acid molecules. In a process called beta oxidation, the alkyl chains of fatty acids are shortened sequentially by blocks of 2 carbon atoms at a time, thereby converting the fatty acids to acetyl-CoA. So here is shown the peroxisomes with the crystallite cores in darkened shade, the organelle microscope. The acetyl-CoA, which is yielded from the fatty acids being broken down in beta oxidation in peroxisomes, is exported, the acetyl-CoA is exported from peroxisomes to the cytosol for reuse in biosynthetic reactions. In mammalian cells, beta oxidation occurs in both mitochondria and peroxisomes. In yeast and plant cells, the essential reaction of beta oxidation occurs exclusively in peroxisomes. An essential biosynthetic function of animal peroxisomes is to catalyze the first reactions in the formation of plasmalogens, which are the most abundant class of phospholipids in myelin. Deficiency of plasmalogens causes profound abnormalities in myelination of nerve cells, which is one reason why many peroxisomal disorders lead to neurological disease. Peroxisomes are usually diverse organelles, and even in the various cell types of a single organism, they may contain different sets of enzymes. They can also adapt remarkably to changing conditions. Yeast cells grown on sugar, for example, have small peroxisomes, but when some yeasts are grown on methanol, they develop large peroxisomes that oxidize methanol. When grown on fatty acids, they develop large peroxisomes that break down fatty acids to acetyl-CoA by beta oxidation. Paroxysms are also important in plants. Two different types have been studied extensively. One type is present in leaves, where it catalyzes the oxidation of a side product of the crucial reaction that fixes CO2 in carbohydrates. This process is called photorespiration because it uses up oxygen, O2, and liberates CO2. The other type of peroxisome is present in germinating cells, where it has an essential role in converting the fatty acids stored in seed lipids into the sugars needed for the growth of the young plant. Because this conversion of fats to sugars is accomplished by a series of reactions known as the Glyoxylate Cycle, these peroxisomes are also called Glyoxysomes. In the glyoxylate cycle, two molecules of acetyl-CoA produced by fatty acid break down in the peroxisome are used to make succinic acid, which then leaves the peroxisome and is converted into glucose. The glyoxylate cycle does not occur in animal cells, and animals are therefore unable to convert fatty acids and fats into carbohydrates. So this reaction is shown here. So you have, let's see, acetyl-CoA. Is that what it is? So fatty acids are converted to acetyl-CoA in animals. Here's shown fatty acid. Or phospholipid, I believe. Plasmalogens are very abundant in the myelin sheets that insulate the axons of nerve cells. They make up some 80 to 90% of the myelin membrane membrane phospholipids. In addition to an ethanolamine head group, over here shown, and a long fatty acid attached to the same glycerol phosphate backbone used for phospholipids, plasmalogens contain an unusual fatty alcohol that is attached through an ether linkage. So we'll continue in the next part.